Okay, team, so today you were introduced to the process of photosynthesis, and as of right now, you know that there are two different components to photosynthesis. So we've got the overall process of photosynthesis, and this can be broken down, as we know, into the light-dependent reactions. We can just call those LDRs, or the light-dependent reaction, and then the light-independent reaction, LIR. And as you know, there are many, many different names for these different things, but they all describe the same reactions. And today we're going to be focusing in on the structure of the chloroplast, as well as the light-dependent reactions. I'm going to give you a little bit of context for what you will be doing in class tomorrow. So let's just dive in here to the structure of the chloroplast. Okay, so imagine here we have a cell, and my whole screen is my cell. And then within my cell, I have a chloroplast here. And now, a chloroplast is composed of, the outside is composed of a double lipid bilayer. So if I were to zoom way, even more in on this, I would see that this is composed of phosphate heads and lipid tails. Oh, this is terrible. But you should know that there's a lipid bilayer here, and then there's another one right here. So this is a double lipid bilayer here. This is terrible phospholipids. But you get the point. Double lipid bilayer, and this is my chloroplast. I'm going to write that to the side. And right here, and then outside of the chloroplast, this would be the cytoplasm of my cell. So the chloroplast is a structure, is an organelle embedded within a larger cell. And then within this double membrane, there is another structure, and I'm going to draw it here. Don't copy me on your notes until after I've finished, and you'll see why in a second, because I'm going to have you erase some things. So inside the chloroplast itself is another membrane system. And this system is composed of sacs that kind of look a little bit like jelly beans. And right here, right now I'm drawing them as independent, but you'll see in a second that they are actually all contiguous or connected. So you'll see why. This actually looks something, like this, where, drawing this too skinny. Each of these membranous jelly beans are, okay, so here you go. All the lines erased and completely reattached. So each of these little jelly beans is attached to every other one, and so this whole system is called the thylakoid membrane system. So this whole system here is called the TH. The thylakoid membrane system. And so this again is another lipid bilayer here, this whole system, the thylakoid membrane system. And each of these little jelly beans is called a granum. So if I were to just box one of these, this is called a granum. and the plural of granum is grana. So a bunch of them. All the grana make up the thylakoid membrane system. And one more piece you should know is that outside of the thylakoid membrane system is the stroma. Now, don't confuse the stroma with the stoma or the stomata that are the opening pore on the underside of a leaf. That's a very common misconception. Okay, so the light-dependent reactions actually take place within the membrane of these thylakoid systems here. And the light-independent reactions take place in the stroma. But today we're going to be focusing in on the light-dependent reactions that are taking place all along these membranes. So I'm going to jump over to another page and draw a picture of one granum right here. Okay, so I've created a granum here, and again for context, this is the stroma outside, and this is the inner thylakoid space, or also called the inner thylakoid membrane. And again, just for context, this is a double lipid, this is a lipid bilayer membrane right here. And so we've got our phospholipid heads would be here, and our tails would extend down into the middle. And so embedded within this membrane are two components of, oh, I forgot to add the A to the stroma, are two components of our light-dependent reactions, LDRs. And these components are called photosystems. So I'm going to draw a photosystem here as a green dot, and another photosystem here as a green dot. And following the photosystems are our ETS, our electron transport system, or ETC, electron transport chain. So the photosystems, their main role is to gather the energy of light from the sun and then pass it off to our ETSs, who, you know, their main goal is to take that energy and bring it down a set of stairs in a controlled way such that we don't kill the baby at the bottom of the stairs, such that the cell can capture the energy within those molecules and harness it to do useful cellular work. It's like your car tank full of gas. You don't just want to light it on fire. You want to slowly combust it so you can put that energy towards useful car work, moving the car forward. The cell wants to do the same thing, transform that energy to useful cellular work. So we've got these photosystems that are going to gather this energy of light and then pass it off to our electron transport chains. And these two photosystems have names. The first one is called photosystem 2. And the second one is called photosystem 1. Now you might think, that's stupid. Why did they name the first one photosystem 2 and the second one photosystem 1? Well, the reason why is because photosystem 1 actually evolved earlier in the history of life. It's the more basic part of photosynthesis. And photosystem 2 was added on later. It's a more complex and further evolved process than photosystem 1. So the work that photosystem 2 is powered to do is different than the work that photosystem 1 powers to do, but the way they gather energy is the same. And I'm going to go on to describe that process in a second. But there's one more thing that you should know here, is that also, let me back up for a second. These photosystems, I've drawn them here, only two on this granum, but you should know that there would be many, many hundreds of photosystem 1s and photosystems 2 all stacked together on all around this membrane. And the reason why this membrane is so folded is to give it more surface area so more reactions can happen here. So if you imagine these two are stacked closely together, there would be another 2 here, and another 2 here, and 2 more here, and 2 more here, and 2 more here, and this is a 3-dimensional structure as well, so there'd be more here, 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 here. So they're all over the place. The thylakoid stacks are just packed with these photosystems and these electron transport chains. The one other molecule that's embedded within this membrane is another protein, and it's a pore. And I'm going to draw it here as a red. I'm going to zoom in a little bit. A red, it's a channel protein. And this protein is going to become, you will see that it is very important. I'm not going to talk about it much today, but over the course of this unit, it will become increasingly clear why this enzyme is so important. So on this enzyme is an active site, and this enzyme is called ATP synthase. And it's powered by a concentration gradient of H+ molecules within the thylakoid membrane that flow down their concentration gradient to phosphorylate ADP into ATP. And so the energy of letting these hydrogens flow down their concentration gradient is going to power this molecule. But more on that later. You should just know that this protein is embedded within the granum as well. It is present. So, taking a step back. Photosystem 1 powers one process, photosystem 1 powers one process, and photosystem 2 powers another process. And I'm not going to tell you what those processes are. That's for a class tomorrow. But I am going to tell you how these photosystems gather energy to pass down to the electron transport chain. And so let's move on to that right now. Okay, so a photosystem is a very complex process, but I'm going to simplify it down for you and make it a little bit more understandable. So a photosystem is composed of pigments, and pigments are organic molecules with a very, very, very important non-organic part. And organic means that they contain carbon, so I'm going to draw this is the containing carbon part of the molecule. It kind of looks like a weird tennis racket here. But this would be the organic part of the molecule. And then in the center is a non-organic part. It is a metal atom. And you know from your days in chemistry, I am sure, that metals are very unique in the fact that they love to lose electrons. And so they are easily, anyone? That's right, oxidized. They love to lose their electrons, so they are always ready to be oxidized. As you guys know from the Chang screencast, the loss of electrons is reduction. Whoa, that was so wrong. Sorry. Take that back. The loss of electrons, I just said it, is oxidation, not reduction. So, metals, they are ready to be oxidized. They're ready to lose electrons. And let's dive in a little bit deeper into the structure of a metal. So you know that a metal atom has a nucleus, which is composed of both neutrons and protons. And then outside of this is going to be circulating electrons. And as you know from chemistry, electrons aren't really circling in a nice circular orbit like planets around a star, but instead they are in orbitals, which are areas of density of electrons, and that with an input of energy, we can bump an electron up from one orbital, a low energy orbital, to a higher energy orbital. And conversely, an electron can bump back down to a low energy orbital, but in the process of bumping back down, it will emit a photon. And I'm going to draw that here as a little yellow squiggle. So, very important. Electrons bumped up, they consume a photon and they bump to a higher energy level, and when they fall back down, they release a photon. Okay, so why is this important? Well, going back to our pigment molecule, this pigment molecule, light from the sun can come over and strike our metal, and this will excite an electron in our metal to a higher energy state. So we've excited an electron here, and so pigments can do this process. And so a photosystem is composed of not just one pigment, but a whole bunch of pigments. And we can draw that structure something like this. So all these pigments are stacked up, one on top of another, or one next to another. But you'll notice that as we get closer and closer to our bottom pigment, we get fewer and fewer per row. So you can think of a photosystem as like a bucket brigade of firemen, each passing buckets of water from one person to the next, except instead of water, pigments are going to be passing energy from one pigment to another. So this is how this whole thing works. Okay, we have light coming in and striking each of these pigments. And when light strikes these pigments, as you know, we excite an electron. So over here in our model here, we imagine the metal ion, or the metal atom within our pigment. Our electron gets excited, it bumps up to a higher energy state, and then as it falls back down to a low energy state, it emits a photon. And this photon passes down from this row, this first row of pigments, to this next row of pigments. But as you'll notice here, there are fewer pigment molecules within this second row. And so we essentially concentrate our photons. So you imagine here, these two pigments molecules are going to pass their excited photons on to just this one pigment molecule here. So we essentially double the energy received by this electron, and which in turn is going to happen in this row, and 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 this row, until you'll notice at the very end we only have one pigment molecule. So all the light that struck these initial pigment molecules, is the energy of that light is passed on to just this one pigment. This guy's so important, I'm going to put a box around him. Okay, so when all this aggregated energy hits this pigment, it not only takes its electron and kicks it up to a higher excited state, but it's so excited that it can just jump right off. So, whee, he loses an electron. It's gone. And now, this is a high energy electron, a he. And this electron can do work. So, this is how a photosystem excites electrons and prepares them to do useful cellular work. And now going back to our picture here, you can imagine we've got all these photosystems here, and they're exciting electrons and then passing them on to our electron transport chain, where they'll be brought down stepwise energetically, releasing that energy to do useful cellular work. And now I'm not going to tell you what that cellular work is, but that will be what we discuss tomorrow. Okay, this was a very long screencast. I apologize. Hopefully this will help you tomorrow. I will end it here now without saying anything more. Oh, shoot, I will say one more thing. Okay, so what you should know that I didn't tell you before, or explain to you before, is that chlorophyll is a pigment. And chlorophyll is doing this work. And we have special names for different chlorophyll molecules, but this chlorophyll molecule is going to be P680, and this one is P700. And that doesn't mean very much to you now, today, but I assure you that it will mean something to you tomorrow. So again, photosystems are composed of chlorophyll. We have different types of chlorophyll. P680 and P700 will be important to you tomorrow. So, okay, that's the last thing I'm going to say.